Hi, and welcome to Science with Mrs. Stone. Today we're going to take a look at seeds. First, gather your materials. You will need a microscope or a hand lens. You'll need some seeds. These pink seeds have been soaked in water for about 1 to 2 days before I started my experiment. I filled up a cup with a small amount of water, added a few drops of red food coloring, and then put the dry seeds into the cup and let them soak for about 1 to 2 days. I also have some dry seeds that have not been soaked in water. A petri dish, and if you want to do the optional extension at the end, you'll need a plastic clear cup and a napkin. Let's start by comparing and contrasting one of the seeds that has been soaked in water. You can see it over here is the pink seed, and a dry seed that has not been soaked in water. Pause the video and take a few minutes to compare and contrast these seeds. Now that you've looked at some of the similarities and differences, it's time to pull the seed apart to find out about the different parts of the seed and what's inside. Carefully peel the seed coat, that's this outer layer, off of the pink seed. Once you peel the seed coat off, you'll notice that the seed will fall naturally into 2 halves. You could try to dissect the white seed, the seed that has not been soaked in water yet, but you'll find that the seed coat is pretty tough and is difficult to pick off. Let's take a close look at our dissected seed. We'll look in very closely right here at this section, and we'll notice this is the baby plant. Let's take a closer look at a diagram. Right here is the same structure we were just looking at. This is our baby plant. Here we can see the baby leaf, and here we can see the baby root. Depending on how long your seeds were soaked, you may notice that that root was starting to poke its way out through the seed coat. The rest of this portion of the seed, everything that you see right here and over here on this half, is all the food source that this baby plant will need to start growing. When a baby plant first germinates, it's not able to do photosynthesis. Photosynthesis is the process by which plants can make their own food using sunlight, carbon dioxide from the air, and water. But before this plant can do photosynthesis, it will need to use the food that's stored here in the seed. The majority of the seed is this stored food. On the outer edge, we peeled this part off. That was the seed coat. What might the seed coat be used for? Here we can take a look at a labeled diagram. Again, we have our baby plant. We have it labeled as the embryonic or baby leaf, the embryonic root, and then this food source right here, scientists call that the cotyledon. And the seed coat wraps around the outside and acts as a way to protect the seed before it starts to germinate. Over here on this side, we can see all of this space is the food source, or that cotyledon, and then it is surrounded by the seed coat. We can use our magnifiers to take a closer look at the embryonic or baby plant. This magnifier is called a Brock Magiscope, and what you do is you take your tray with your dissected seed and you put it on the stage of the microscope. That's this flat part right here. And then you take this tube and you twist it up and down while you're looking through the top in order to get it into focus. When you get it into focus, you should be able to get a good look at that baby plant. Here I can see the embryonic leaf right here, and here is the embryonic root. If you want to watch a plant grow over the course of the next few weeks, you can take the plastic cup and the napkin, and you want to wrap the napkin inside the plastic cup so that it's pushing up against the sides. Then you want to take a few seeds that have not been soaked in water, and you want to tuck them in between the napkin and the plastic cup. You can see one seed over here. I have another seed tucked in over here. Then you'll put some water into the cup. Not a lot of water, you just want to put a little bit in the bottom, enough that it will start to wick its way up the paper towel. And you want to make sure that this paper towel stays wet. That will allow the seed to start to germinate. And over the course of several weeks, you'll be able to see these changes occur in the seed. The first thing that you'll notice is that baby root will start to poke its way out through the seed coat, and work its way down. The root continues to grow, and then the seed will start to push its way up. If we had this in soil, it would push its way up through the top of the soil. Since we don't have this in soil, it will just start to push its way up towards the top of the cup. And you can see here, this is the cotyledon, or that food source, and then we can see the baby leaf starting to poke its way out through the top. As the plant continues to grow, we'll see those leaves get larger, and you'll still see a little bit of the cotyledon, or that food source. That food source will start to shrivel up as the plant uses it up. Once this plant grows its second set of leaves, then it will be able to do photosynthesis, which is how it makes its own food. Be sure to keep your paper towel wet during this process so that your seed will continue to germinate. Enjoy the activity.